Adult zebrafish has a unique ability to regenerate neural system after injury, such as hair cells, olfactory bulb, telencephalon, spinal cord, and optic nerve. As a part of CNS, optic nerve injury could induce RGC survival and regeneration. However, HE staining and antibody IHC could not specifically mark RGCs, with transgenic line marked all RGCs has not been constructed. In order to research the change of RGCs number after optic nerve injury in adult zebrafish, we successfully labeled all RGCs by retrograde labeling RGCs from the tectum.
Hi, I'm Chen Tian.
I'm Tian.
We are members of the Laboratory of Neurodevelopment and Repair, School of Life Science, University of Science and Technology of China.
And today we will show you how to retrograde labeling RGCs in adult zebrafish and get the RGCs number in whole mountain retina.
So, let's get it started.
First, we need to build up a perfusion system to perfuse anesthesia and fresh water. You can find more details about the apparatus in the attached PDF document of this movie. Put the box above and make sure the fluid can flow smoothly from the box to the fish. Keep all apparatus disinfected during the surgery. Prepare several spitballs and put them in the caves.